Hello everyone and welcome to today's webinar, Water Quality for Pharmaceutical and Medical Device Processes, brought to you by Nelson Laboratories in partnership with QMed. I'm Tanya von Grumkow, Program Manager at UBM Americas, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. Each widget within the dock at the bottom of the console will open in a different window for you to view additional resources, speaker bios, and more. There are several social media widgets for you to like, share, and talk about the content with your peers and communities. At the end of the program, we will ask you to complete a short survey. Please take a moment to fill this out. Your feedback will provide us with valuable information on the subjects covered in this webinar and how we can provide improvements in future events. You can also launch a survey at any time by clicking on the survey button at the bottom of the console. The slides will advance automatically throughout the event. You may also download a copy of the slides by clicking on the PDF labeled Download Slides, located in the Additional Resources widget. If you're experiencing any technical problems, please visit our webinar help guide by clicking on the Help widget. Now on to the presentation. Discussing today's topic will be Paul Littley, Consulting Manager at Nelson Laboratories. Paul, I now hand it off to you.
Thank you, Tanya. Welcome, and thank you for joining me today for this presentation on water quality for pharmaceutical and medical device processes. As Tanya stated, my name is Paul Littley, and I'm a consulting manager with Nelson Labs. I have over 11 years of high purity and quality water system design, validation, process controls, and U.S. bulk water generation. I'm excited to be involved in this webinar today, presenting on one of my favorite topics and sciences I am very passionate about. We start off today with the main question of why water quality. Why is water quality so important? Well, water is arguably the most widely used raw material in medical device manufacturing, pharmaceutical processing, biotech, and tissue bank processes. The selection of the appropriate water quality or water grade is critical for many reasons, such as reduction of patient risk to infection and contamination control in the manufacturing environment. When we use the term water quality in the medical device industry, we are typically referring to water quality or purity of water greater than that of potable or drinking water from a municipal source. The quality of water is dependent on the manufacturing process, product need, and regulatory requirements. Contaminants such as bacterial endotoxin, microbial contamination, and particulates from water of lesser quality used in the cleaning process may significantly impact the health of the patient, the functionality of the device, or the performance of the manufacturing process. General uses of water range from active pharmaceutical ingredients to dosage forms for parenteral drugs, analytical reagent mixing, and bridge water for cleaning process, as well as chilled water for extrusion processes. As I stated in the opening slide, poor water quality or residual impurities in the water have the potential of adversely impacting the product, process, or even patient health. When selecting water quality for a given process, this following statement is truly, in my opinion, summing up the need or requirement for water quality in the manufacturing process. This statement, the level of acceptable impurities is determined by the potential of that impurity to negatively impact the product, process, or patient health. The understanding of potential impact is key in quality water system design, validation, and process control. In today's presentation, we will discuss regulatory background, water basics, such as categories of impurities found in water and water chemistry. We'll also discuss approaches for water quality selection, define critical quality attributes, and talk briefly of water system design. In closing, we will cover water system process controls, preventive maintenance, and system monitoring. So to dive in, we'll start with regulatory requirements. For pharmaceutical uses and purposes, the regulatory requirements are fairly straightforward with regards to water quality. We have the FDA, United States Pharmacopoeia, European Pharmacopoeia, Japanese Pharmacopoeia, as well, for example, providing regulatory requirements for water quality. However, when we move into the realm of non-compendial waters, specifically water for medical device reprocessing, guidance becomes significantly vaguer. The onus of proper selection criteria for water quality is the responsibility of the manufacturer, with the expectation that an appropriate water quality or water quality grade for the process or products will be produced. Industry-related regulatory requirements and in some cases guidance can be found in the following documents: CFR Part 210, CFR Part 211, Part 820 for medical device, as well as the USP 1231, and even the document that FDA posted in 1993, which is still relevant, the Guide to Inspection of High Purity Water Systems. And in closing and final, you can also look at AAMI TIR 34, water for reprocessing of medical devices. To exemplify some of the vague nature of the CFR, I've copied the definition from Part 210, where it lists a component is any ingredient intended for the manufacture of a drug product, including those that may not appear in such drug products. This is a general application of the definition for an API, for example, but keep in mind it doesn't have any direct reference to pharmaceutical waters or the quality of water required, nor does it talk about potential impurities that may harm the patient. In this slide, we have the definition of manufacturing material from 21 CFR, Part 820, the QSR. A manufacturing material means any material or substance used in or used to facilitate the manufacturing process of a concomitant, constituent, or byproduct constituent produced during the manufacturing process, which is present in or on the finished device as a residual or impurity, not by design or intent of the manufacturer. Again, general regulation of a manufacturing material. So the takeaway with the regulatory interpretation is that water quality is regulated regardless of the regulatory claim. It is expected that the processing of water used in the production process be free of objectionable contaminants based on the needs of the process, and the manufacturer must identify and develop suitability of water used in the manufactured process, and of course, application of the CFR requirements to your product. In closing the regulatory topic, I always like to advise my audience not to take my word for it. The evidence is in the public domain. Here we have our first warning letter that we'll look at, which is specific to microbial control of water used as an API, active pharmaceutical ingredient. For this firm, the FDA cited that failure to ensure the water was appropriate quality when used in the final purification steps of active pharmaceutical ingredient, USP grade, intended for the use of a parenteral drug. And furthermore, procedures for controlling and monitoring of the water system were not established or qualified. Obviously, they had levels of microbial contaminations that were far beyond the levels required for the pharmaceutical ingredient that they were mixing. This next letter is specific to process controls as well with regards to microbial and endotoxin excursions. The inspector identified quickly the randomness of the system sanitization and lack of system control on part of the manufacturer. Clearly, this is a breakdown in the manufacturer's quality system with regards to process control, as they had infrequent and redundant disinfections of the system multiple times throughout the years reviewed. Moving into the next section of today's presentation, we look at water impurities and basic water chemistry. Simply stated, if it is not hydrogen or hydroxide, it is an impurity. Again, let me reemphasize that the level of acceptable impurities is determined by the potential of that impurity to negatively impact the process, product, or patient health. In this slide, we see a supposed example of visually poor quality drinking water from Sochi, Russia. Note the discoloration, which we commonly refer to as turbidity, or more accurately, undissolved suspended solids. Water is an extremely aggressive solvent and commonly referred to as the universal solvent. Over time, water will dissolve almost any substance to which it is exposed. Due to this fact, even potable or drinking water from a trusted municipal source will contain a high level of impurities absorbed from the environment. This slide is a perfect illustration of water's ability as a solvent. If you've ever been fortunate enough to see the Colorado River up close at the bottom of the Grand Canyon, you'll know that this picture is not altered or photoshopped, and that is the true color of the water. We commonly categorize water contaminants into 5 major groups: suspended particles or undissolved solids, which can visually be observed as in our previous slide. Next are the ionic impurities. These are the soluble solids categorized as total dissolved solids, or TDS, such as salts, metals, and other anions and cations. Microbial impurities, such as microorganisms and endotoxins. Organic impurities encompass those organic compounds and tested as total organic carbon, or TOC. And lastly, we have the dissolved gases, typically from atmospheric sources. Sources of the impurities span both the natural environment, but also man-made chemicals and even byproducts that have worked their way into the watershed over time. Let's first start with suspended solids, which are also referred to as turbidity, visual cloudiness, and clarity of the water as previously mentioned. These suspended or undissolved solids include silt and clay particulates, plankton, algae, fine organic debris, and other particulate matter. This pictorial representation shows a range of water with low turbidity on the left to water with higher turbidity on the right. Suspended solid particles typically range in size from sand at 1.0 millimeters down to clay and silt at 0.004 millimeters. These particulates will typically not pass through a filter with an active pore size of less than 2 microns, or 0.002 millimeters. Total dissolved solids, or TDS, refers to mineral salt and metal ions such as calcium, chlorides, nitrates, phosphates, iron, and sulfur. These are cations and ions and anions, which are polar molecules and therefore soluble in water. These are molecular size impurities that will easily pass through filters with an active pore size greater than 2 microns. Drinking water from municipal sources will typically measure less than 400 parts per million, but depending on the area of your facility as well as the season, these total dissolved solid levels can be significantly higher. Turning to microbiological impurities, fecal coliforms such as E. coli and Enterococcus are sources ranging from animal to human waste and are a significant concern not directly but indirectly as they are indicators of potential presence of associated pathogenic organisms. If coliforms are present, action should be taken to ensure the water system is capable of removing these microbiological impurities. In this slide, we have listed a few of the more common waterborne organisms. Pseudomonas species are very common in water, as well as Burkholderia, Roseomonas, Bacillobacterium, Deinococcus, Ralstonia piquete, and Brividomonas dementia, as well as other gram-negative organisms. Continuing on in the microbiological impurity category, bacterial endotoxin are, of course, a major concern of common waterborne contaminants. Endotoxins are constituents of the gram-negative bacteria cell wall with the capability of inducing pyrogenic immune responses such as fever and nausea. As we covered in the previous slide, gram-negative organisms thrive in aqueous environments, and therefore the risk of endotoxin impurities presents itself quite readily. Next, we move to organic impurities, those impurities that are carbon-based and sourced from plant and animal decay. They may also be from industrial and domestic waste sources such as pesticides and herbicides, and basically runoff that accumulates in the watershed. We quantify the levels via total organic carbon, or TOC, testing. And finally, the last category we will discuss is the dissolved gases. These are typically atmospheric gases such as carbon dioxide, oxygen, nitrogen, but also ammonia that can accumulate in the water system. Moving into water chemistry, we'll briefly discuss conductivity, resistivity, and pH. Electrical conductivity in water is directly related to the ionic content, and therefore it's affected by the presence of inorganic dissolved salts such as chloride, nitrate, sulfate, and phosphate anions, which carry a negative charge. It is also affected by sodium, magnesium, calcium, iron, and aluminum cations, which carry a positive charge. Technically speaking, water conductivity is a measure of the ion-facilitated electron flow through water. More clearly stated, it is the ability of water to conduct an electrical current based on its ionic concentration. In this graph, we see the relationship between electrical conductivity and total dissolved solids. As TDS, or ionic concentrations, increases, so does electrical conductivity. Because of this relationship, we're able to use conductivity as an indirect measurement of the ionic concentration existing in the water sample. This next slide is an excellent comparison of the ionic purity of water grades versus conductivity. Conductivity has the units of microsiemens per centimeter, and we can see that the theoretical conductivity of pure water is 0.055 microsiemens per centimeter. An approximate determination of the ionic concentration can be made by the conversion of 1 part per million TDS, approximately equaling 2 microsiemens per centimeter squared at 25 degrees C. This conversion is based on the sodium chloride salt conversion. It should also be noted that we sometimes use the term resistivity when discussing ionic concentration. Resistivity is a measurement of the resistance of water to an electrical current. Resistivity is simply the inverse of conductivity and has the units of megohms, centimeters. As we know, pH is a measure of acidity and or basicity of an aqueous solution, and more precisely is the measurement of the hydrogen activity of the solution. Pure water has a pH very close to 7 or neutral. pH is typically a relevant measurement when analyzing high-purity water systems, as it requires significant levels of TDS or dissolved gas to shift the scale one way or the other. Because of this fact, the USP does not have requirements for pH. In the next section, we will discuss selection of water grades or water quality, which is the basis for system design. Water quality standards are developed as a three-part system for protecting the product quality and maintaining system integrity. This three-part approach to proper selection of water quality includes defining the designated use by identifying what the water will be used for and what impurities should be removed. Next, the firm should identify water quality criteria to be measured, such as microbial, TOC, or conductivity levels, and determining acceptable levels required to meet the designated or intended use of the water. Lastly, establishing an anti-degradation strategy to maintain performance and the longevity of the system. The anti-degradation strategy should include proper design, proper operation, and proper process controls to minimize biofilm formation and system fouling. A few common categories of water grades are listed here in chart form. On the left-hand side, we have the water grades or title of the water quality, and then we have which agency has designated these titles. And on the left and the top, we have non-potable, non-drinkable water, which the EPA has categorized, but also potable drinking water under the EPA, as well as utility water by AAMI out of TIR 34. TIR 34 also lists critical water, which is a higher quality of water for reprocessing medical devices. The next two grades are USP and EP grade waters, and we'll discuss these a little bit later in the presentation. We'll also see that there's a highly purified category, water for hemodialysis, and sterile purified water as well. As we discussed in the beginning of the presentation, compendial requirements are well defined in general information section of the USP under section 1231, water for pharmaceutical purposes. This is a portion of figure 2 from section 1231 and is a basic decision tree to assist the user determine the most appropriate water quality for the intended use. As we also discussed earlier, there are a few aids to assist the user with regards to non-compendial water. In this slide, I have represented this chart from 1231, but the intended use with rinse water, equipment rinse water, or other obviously do not carry designation or assistance from the regulatory community. Because of this limited information in our industry, prior to system development and planning, it is crucial to have a sound knowledge of water quality and water systems, as well as a basic understanding of product design and intended use of the water. Awareness of the production processes associated with the product manufacturing environment is also crucial. If the firm does not have this level of expertise, it is highly recommended that the manufacturer partner with a water system engineering or design firm for consultation. Moving into the next section, we'll discuss critical quality attributes commonly monitored to verify system performance. When we speak of critical quality attributes, we're speaking of a physical, chemical, biological, or microbiological property or characteristics that can be defined, measured, and continually monitored to ensure final product outputs remain within acceptable quality limits. This is not to be confused with critical process parameters, which are parameters whose variability has an impact on the critical quality attribute and should therefore be controlled and monitored as well. To further clarify a critical process parameter in water systems, for example, parameters such as flow rate, temperature in hot systems, and filter pressure differential should be controlled and monitored. These have a direct impact on the critical quality attributes of that system. The four most common critical quality attributes in water systems are conductivity, which is measured and monitored to ensure the ionic impurities are within acceptable levels. Total organic carbon, which is measured and monitored to ensure the organic impurities are maintained within acceptable levels. Microbial content, which is measured, monitored, and in some instances even speciated to ensure the microbial load of the system is within the acceptable levels. And lastly, depending on the quality of water or grade of water being produced, endotoxin levels are measured and monitored to ensure that the impurities are within acceptable levels. In the next two slides, we'll look at two examples of acceptable critical quality attribute levels for purified water and water for injection per the USP and EP, European Pharmacopoeia. The critical quality attributes are microbial load, conductivity, TOC, and endotoxin. And as you can see, are fairly harmonized between the two branches. Although microbial load is not directly referenced in the monograph for the USP, it is implied that an action level of less than 100 CFU per mil be instituted. Also, another difference is the endotoxin levels between USP are not required, where EP they are required at 0.25 IU per mil. And IU is an international unit, which is equivalent to a endotoxin unit. Looking at water for injection specifications, again, they're fairly harmonized. Again, the microbial loading is implied less than 10 CFU per 100 mil. And again, the rest of the critical quality attributes between conductivity, TOC, and endotoxin are harmonized fairly well between the two agencies. Moving into our next section on water system design. Design is not only crucial to meet the designated use requirements, but also to maintain the system performance throughout the production life cycle. Improper design or design flaws have the potential to plague a system and significantly impact the quality of water and, in turn, the quality of product. In this slide, we see a generic reverse osmosis deionized system with storage tank and distribution loop. Incoming feed water from municipal sources is first entered into a sediment filter, which will remove suspended solids and turbidity. The water then goes into a softening tank, which removes calcium and magnesium elements, those hard scaling minerals, from the water. Then the water is treated with a carbon filter, carbon filtration, which removes chlorine and some total organic carbon. It is important to remove the chlorine prior to the RO membrane, as chlorine can damage the central membrane of the system. After RO, the water typically goes into a storage tank or is treated with deionization. Once in the storage tank, it goes into the distribution loop for use. It is not uncommon for additional DI and even a UV sanitizing lamp prior to distribution to be in place. The minimum requirement or quality of feed water per the USP and World Health Organization is drinking water quality. This is important when designing any system. If you're sourcing water from a municipal source or from a well that does not meet drinking water quality, you must treat it to the level of drinking quality before it actually enters into your water system purification process. Common feed water pretreatments reduce the feed water variability by reducing the incoming impurities. These are typically inexpensive components and therefore reducing the maintenance costs on the higher end components such as the RO membrane and the distribution loop. A lot of the methods involve depth filtration of 5 micron filters or less, multimedia filtration, as well as softening and anti-scaling chemicals, as I was discussing in the previous slide. Looking at the mechanism of osmosis and reverse osmosis, osmosis is the natural diffusion of water across a semi-permeable membrane from an area of low salt concentration to an area of high salt concentration. Reverse osmosis reverses this process using pressure to force the water across a semi-permeable membrane in order to remove salt and other dissolved solids. RO membrane filtration typically rejects 90 to 95% inorganics, 90 to 95% organics, 100% bacterial, and 100% endotoxin impurities. The ionization of water is an ion exchange process which converts salts and acids to hydrogen cations, and then those cations are removed by anion exchange. It removes a good 95 to 99% of the inorganics remaining in the water after the RO has already removed 90 to 95% of those initially. Technologies for deionization include cation and anion beds, as well as mixed beds, and even electrodeionization processes. Water storage and distribution considerations. Due to the fact that RO makeup of water is very slow, accommodation for water storage conditions should be made to meet peak demands, as well as account for production expansion. Also, considerations should be taken that the water storage is an area of reduced flow, as well as the water storage will require DI treatment and microbial control prior to distribution. With regards to the distribution loop, Considerations of hot versus cold loop distribution should be considered, as well as the piping material and flow rates. Point of use valve configurations should be carefully considered as well, as the inappropriate or improper valve might be linked to fouling at that point of use. When we talk about microbial fouling, we typically talk about biofilm formation and propagation. Biofilms will occur and they are virtually impossible to eliminate. However, biofilms can be controlled through design as well as proper maintenance and process controls, such as increasing the flow rates or the shear force within the pipe wall to cut down or restrict the organisms from actually adhering to the pipe wall, as well as routine sanitization if the system is a cold water system, control of the total organic carbon levels, as well as design. Designing against dead legs and low flow areas are key to reducing the chance and formation of a biofilm. When we talk about biofilms, we talk about typically two categories of biofilms, if you will. We talk about a hard and secure or a loose and fluffy. The hard and secure biofilms will be very difficult to remove, but also they won't shed as much. A loose and fluffy biofilm has the potential of breaking loose and propagating throughout the entire system. The measures stated above, increased flow rate, routine sanitization, and such, will reduce the chance of having this loose biofilm formation, and thus, therefore, reducing the chance of the biofilm propagation throughout the system. Looking a little bit more closely at biofilms, this slide is an excellent example of how biofilms actually form and establish themselves. We see that biofilms can adhere to the pipe wall very quickly and start to begin to propagate, as well as build nutrients and then collect other organisms, as well as nutrients as they grow and establish themselves into a well-established colony. When talking water system design, we have several rules of thumb to follow. First, we want to prevent and design against dead legs and low flow areas where these biofilms can form. We want to maintain a turbulent flow environment versus a laminar flow to maintain a shear force to keep the organisms or potential organisms from adhering to the wall and establishing biofilms. We also want to reduce our TOC levels to minimize available nutrients to these organisms, as well as incorporating a robust sanitization strategy. Designing for longevity or overbuilding for the system is key as well. It comes down to spend now or spend later. Costs associated with component replacement and even requalification and downtime can skyrocket. Design with the end in mind and consult early. Moving into our final section of today's presentation, we now talk about water system process controls. With regards to water system process controls, applying best practice will pay dividends in the end to maintaining a lifelong use for a water system. Maintaining the quality water requires system monitoring and training, as well as correct interpretation of those test results. Establishing a robust sampling plan and associated baseline is vital to detection of potentially catastrophic impacts, which will lead to or result in downtime and costly replacement and requalification costs. With regard to routine monitoring, this is where we're talking about testing and training of those critical quality attributes that ensure the system will function as designed. Sample plans, as always, should be documented, trended, and periodically reviewed, as well as adjusted to ensure the system performs at its optimal level. Action alert levels should be established and applied to monitor and trended results to ensure early detection of trending or out of spec conditions. Alert and action levels are distinct from process parameters and product specifications. They are used for monitoring and control rather than accept or reject decisions. Alert levels are levels or ranges that, when exceeded, indicate that a process may have drifted from its normal operational condition. Alert levels indicate a warning and do not necessarily require correct information. However, exceeding an action level indicates that corrective action should be taken to bring the process back into its normal operating range. Process controls are not limited to monitoring and trending of data. Adherence to a strict preventive maintenance program is of equal importance in maintaining the operational state of a high-quality water system. Water system maintenance should never be scheduled on an as-needed or reactive basis. Daily, weekly, and monthly preventive maintenance plans, such as measuring of differential pressure across filters to see when they're being saturated. Daily measurements of conductivity trending, typically from an online meter, are also recommended, as well as measuring the TDS from incoming seawater. It's also not a bad idea to measure your RO permeate, or the output of your RO, to establish a baseline for RO performance with regards to total dissolved solids. And of course, system sanitization, typically on a quarterly basis for cold water systems, is the best practice as well. Quarterly and annual preventive maintenance plans would also include DI bed replacement or rejuvenation, pre-treatment component flushing and rejuvenation, such as the water softening and carbon beds, system sanitization, depending on your system performance. If quarterly is too frequent, you can move it to a semi-annual as well, and RO unit inspection and servicing. On a quick safety note, high purity water is considered to be unsafe for human consumption. It is considered hypotonic, which refers to a decreased solute in the solution, or in the case of ultra-pure water, a demineralized solution. Consumption of quality water would cause the body cell to lose electrolytes and cause dehydration-like effects. And refer back to the osmosis slide that we discussed earlier with regards to how osmosis works versus reverse osmosis. This brings us to a close of today's water quality presentation. In summary, the goal of effective water treatment in the MedTech industry is to prolong the life of medical instrumentation, ensure the effective device functionality, and to minimize the risk of adverse patient outcome arising from contamination. Management of a quality water system is paramount to every healthcare facility and/or manufacturing environment. It is the responsibility of the manufacturer to ensure the proper water quality is selected and maintained for the intended use as a raw material in the manufacturing process. Thank you again for joining us today for this water quality presentation. And now, back to you, Tanya.
Thank you, Paul, for providing the valuable information on water quality for pharmaceutical and medical device processes. We appreciate your time with us today. For upcoming webinars and for the archive of this webinar, please visit qmed.com. This webinar is copyright 2015 by QMed. The presentation materials are owned by or copyright by Nelson Laboratories. The individual speaker is solely responsible for his content and opinions. On behalf of QMed and our sponsor, Nelson Laboratories, thank you for attending and have a great rest of your day.